Hi, this is Alex from devstories.eu. Scanpy Single Cell Analysis, episode 16 of 16. What happens when your single cell dataset hits 5 million cells and completely exhausts your computer memory? You cannot filter it, you cannot normalize it, and your kernel simply crashes. The solution to this hard memory limit is experimental scale-up with Dask. People often hear Dask and immediately think of parallel processing across a distributed cluster to speed up code. While Dask can do that, its primary superpower in Scanpy right now is out-of-core memory management through lazy evaluation. It is not just about doing things faster, it is about doing things that were previously impossible on a single machine. Standard Scanpy workflows rely on in-memory arrays. This requires the entire dataset to reside in your active RAM. When you use the Dask backend, Scanpy replaces these standard arrays inside your AND-data object with Dask arrays. A Dask array is essentially a collection of many smaller arrays called chunks. Instead of loading the whole matrix into memory at once, Dask leaves the bulk of the data safely on disk. When you run a Scanpy pre-processing function backed by Dask, it does not calculate the result right away. This is where lazy evaluation comes in. Instead of crunching numbers, Dask builds a recipe. It creates a task graph outlining exactly what mathematical operations need to be performed on each individual chunk of data. Consider a scenario where you have a massive 2 million cell dataset on disk and you need to calculate quality control metrics. If you try this with a standard array, your system will freeze as it tries to pull everything into RAM. But if your AND data object contains a Dask array, you simply call the standard Scanpy quality control function. The function returns almost instantly. Your memory does not spike because no numbers have actually been processed yet. Scanpy merely noted your intent. When you are finally ready to plot those metrics or save the summary statistics, you explicitly tell Dask to compute the result. This is the part that matters. At this exact moment, Dask pulls the first chunk of data from your hard drive, calculates the metrics for just that chunk, stores the small result, and then immediately throws the raw data chunk out of memory. Then it moves to the next chunk. Your octave memory footprint stays tiny. It is dictated entirely by the size of a single chunk, not the millions of cells in the full dataset. Right now, this Dask backend in Scanpy is categorized as experimental. Not every function in the ecosystem supports it yet. However, core pre-processing steps like normalization, scaling, and highly variable gene selection are fully equipped to handle massive out-of-core operations. When you shift from eager processing in memory to lazy processing on disk, the size of your dataset is no longer limited by your hardware RAM, but only by your patience. This brings us to the end of our single cell series. I highly encourage you to explore the official Scanpy documentation and try building these data graphs hands-on. If you have ideas for what technologies we should cover in our next series, visit devstories.eu and let us know. Thanks for listening. Happy coding, everyone.